First of all, I'd like to thank Muriel and Hervé for this invitation, for the chance to meet you all. And I thought the program of the course was amazing. That's why I decided to stay the entire week. And I'd also like to thank the other presenters, because the lectures were very well articulated and indeed very interesting. So a bit about myself. I'm sort of a jack of all trades and master of none. I studied engineering in Argentina, where I grew up. Then I moved to the US. At Cornell University, I studied applied physics and planetary physics. That's what I did my PhD on. And a bit about origin of life studies. Since '92, so that's about 25 years ago, I've been working at ESA, but on different things. I've moved from human spaceflight to Russian capsules to parabolic flights, things on the space station. And then for the past 15 years or so, I've been working on trying to put a very nice rover on the surface of Mars, which I will describe on the second part of my talk. So you have to bear in mind my limitations. So I'm sort of engineering/physicist, but what we have to deal with here has also a lot to do with chemistry. Now, luckily, I married a chemist, so that helps a lot. So Katerina, my wife, is a peptide synthesis chemist, and we have very lively discussions at home, where I tell her that everything is physics, and she tells me that we physicists don't know much. We only understand 4% of the matter in the universe, and that at any rate, anything that is worth knowing anything about is chemistry. So it's always fun at home. One of the disadvantages of being the last lecturer is that you don't know what it is that the other guys will talk about. So you're going to see that there is a bit of overlap with some themes that have been discussed already. Now, I could have taken those view graphs out, but there is a reason why they're there. And bear in mind when I go through the talk that the main reason why I discuss these things is because we are trying to put together a space mission. And so from all the abstract concepts, we have to distill something that gives us practical recipes for building something and operating it on another planet. So you may agree or disagree with some of the things that I will say, and in some cases you may think that they are gross oversimplifications. But the goal is to try to collect what we need in order to build something that is relatively crude, which is what a space mission is, and do some science with it. So the way I've structured this is, first of all, there's a bit of science interest and pretty pictures at the beginning. And the reason for that is that initially Muriel asked me to talk about solar system exploration, but of course I quickly realized that this is way too vast a subject. So then I decided that I would only talk about those missions that have something to say about exobiology. So I will disregard everything else. No plasma physics, no surface geology, just touch on those things that help us in order to define ExoMars, which is the second part of the talk. So the first part is all the blah blah that gives you the Lego bricks for then trying to decide what you're going to do to put this mission together on the second part. So I'll start with a bit of history, but it's only one slide. And that is, our place in the universe has changed and keeps on changing. So how we see ourselves. In the beginning, and in ancient Greece, the discussions about where humankind was and the Earth was in relationship to the cosmos was a bit of a philosophical thing. I like the concept of Epicurus very much, which was one of the first, not the only ones, who came up with this idea of the infinity of worlds, but he did so from a relatively a philosophical point. Basically, the Greeks invented this concept of the atom, which of course they couldn't see. For them it was an abstraction. They said there must be something that is the smallest thing you can have. We'll call it atom. And since there's gazillion of them, there has to be gazillions of the big things that the atoms are used to build stuff. And so just from those arguments, this concept of there must be more planets came about. Now, Ptolemaeus was a Greek who happened to live in the Roman province of Alexandria in Egypt. You know, the Roman Empire was a bit everywhere that counted for something in the ancient world. And basically, he was looking at the planets and tried to come up with a concept that would explain what the planets were doing. So he invented these funny orbital motions in order to explain how the Earth could be at the center of everything and still sort of explain how the planets moved. And for many centuries that worked a treat, because you could predict many things using his model. Copernicus proposed this idea that the planets actually orbit around the sun. But you see, now we have moved forward quite a bit in time. And after, if you want, the dark ages of medieval times, my son who studies history would disagree with that. He doesn't think that the medieval times were so dark. But most of the knowledge was in the realm of the church. And the church was a very important organization, had a lot to say also in government and what the future or not of budding scientists would be. So Copernicus was very careful to publish this work just the last year before he died. So he sat on it for a long time. But it was simpler from the point of view of understanding the motion of planets, and it also worked very well. We heard about Giordano Bruno in the lecture, who came up with this intuition, if you like, that stars are suns. He was a contemporary of Galileo, and both him and Galileo were sort of universal men. They had been everywhere. Giordano Bruno taught in France, in Germany. He was in England as well. And in the end, both him and Galileo applied for exactly the same position at the University of Padova. In the end, Galileo got the job. And Giordano Bruno, while he was waiting to learn whether he had been accepted to become a professor at Padova or not, he had a bit of the bad luck to start teaching in Venice one of the important persons from one of the Doge families of Venice. And they got into a dispute. And it was him that got him into this anathema process that eventually got him into the big mess. The process started in Venice, then he was transferred to Rome. The whole trial lasted almost 7 years, and you know that in the end he was burnt. So he was, if you like, a victim of more of an influential family not liking what he had to say so much, but not necessarily had so much to do about the actual things he was proposing. Now Galileo, as we learned, started using the telescope, but to me the most important thing he did was the work on the moons of Jupiter, because there, for the first time, you had a practical demonstration of a mini system turning around something that was not the sun. So this was a very powerful proof that indeed this concept that there may be other worlds turning around other stars and even moons turning around planets became more firm. Then in a very short time we had some very important works, like the work of Kepler that determined that planets moved in elliptical trajectories, and then Newton that established that gravity is universal. And since then, for all this time, we have slowly found out that there are such things as universal laws and that they apply everywhere. But so one could simplistically say, well, if the same laws apply everywhere and we got life here on Earth, there must be life in many other places. But it is not that clear that even if the same laws apply, we get the same effects everywhere. And we will see that, or you have seen already during the conference, that this is indeed the case. So now I have 10 or 12 slides in which I just show you pretty images. And some are new, some are old. Like, for example, these are the first, one of the first images of Mars taken by Mariner 4 in the 1960s. Here's a whole bunch of other missions that contributed to what we know, particularly early ones. One of the things we are seeing now is that more and more amateur astronomers or people who do not have a scientific background but are really, really interested in what comes out of the missions have started to contribute. And in some cases, they do really nice work. For example, these are some of the Venera images. There were several landers. This is Venera 13 of the surface of Venus. And these cameras had spherical projections. But recently, one of these amateur astronomers did a very nice reprojection of the original images, and you can see, well, maybe you cannot very well here, but you can on my screen. You can have a very nice being there view of the surface of Venus. You see the basaltic plains. Venus is a pretty unlucky planet, unlucky also for us from a science point of view, in that the surface has been recovered by relatively modern lavas about 600 million years ago. So it's very difficult to understand something about the early history of Venus, even though it may have been as interesting as that of the Earth and Mars. Recently you have seen the images from New Horizons. These are sort of tweaked from a color point of view. I prefer to look at them in a true color. But this has been such a surprise. What a rich set of moons that Pluto has. Who would have thought? And some with these funky motions, sort of pseudo-stellar oscillatory random motions. There's several of them. Another mission that has provided us with a great wealth of data and also amazing images has been Cassini-Huygens. And in Europe we have much to be proud of. Here you see an image of Titan with the landing of Huygens. This is the classical image, and is the only one we have of the surface, because actually Titan is pretty dark. So this was the bit of surface that was being illuminated by the one single light bulb lamp that the lander had. So after it touched down, it had this little lamp providing a swath of light on the surface, and that's what you see. There have been many very nice images coming out of Cassini, and if you go to their site, some are, I don't know, inspiring. You could almost say artistic. Very pretty. One of the major discoveries of Cassini, and some of you are working on this, have been the plumes coming out of Enceladus. And this is a view graph that a colleague of mine produced that I like quite a bit. Here you see different albedos of objects in the solar system. So you see here Enceladus, which is very reflective. It has an albedo of 1. The Earth, which has a relatively low albedo because the sea is blue, is very dark, absorbs a lot. And the Moon, who we see on the sky as being white, but is actually pretty black. And then you can see the difference in size of the objects, which is something that is very, very important, as we will see, for the capacity of an object to retain or not an atmosphere, and for how it is structured internally. But it's not only the planetary missions. Hubble has also given very nice results in the planetary domain. For example, this is a view of the surface of Jupiter, and with that they produced a very nice movie of the zonal circulation of the upper atmosphere of Jupiter. There has been Rosetta. I'll just show 3 or 4 pictures. This has been another amazing achievement. Besides what we have learned about the comet, learning to fly a spacecraft around such a low gravity object has been very, very interesting. It turns out you cannot go into orbit about a comet, so you end up flying high energy hyperbolic segments, which you have to control very carefully. And on top of that, the comet is an object that this comet spins relatively fast, as you can see here, and with a non-uniform, to say the least, gravitational field. So it's been very interesting. The shape of 67P was a surprise. And we saw in Alessandro Morbidelli's presentation how these comets are interesting because their composition records something about what the very early solar system volatile composition was like billions of years ago. And so many comets are stored in these two main reservoirs. You have the Oort cloud and you have the Kuiper belt. And every now and then, one of them will have its orbit destabilized and will be thrown into the inner solar system. And that's the end, if you like, or the beginning of the end of the life of the comet. Because as it gets close to the sun, it starts losing matter. Its surface is reworked, and it loses in the order of a meter of its outer shell per pass around the sun. So depending on the initial size of a comet, it will last a few tens of orbits, or it may last hundreds or thousands of orbits. But when you study a comet, of course, you don't have access anymore to the pristine, let's say, composition or arrangement, maybe not composition, of the elements of the comet, but you have to deal with the fact that the surface has been reworked. Nevertheless, they are very interesting objects. You see here some views of the exquisite detail that was possible to get with the Osiris camera on the Rosetta spacecraft. This is something I like very much. Is eventually we got to catch a glimpse of how the plumes in the comet were coming out, the ones that then give rise to the coma. And so here you see sort of an outburst from the surface of the comet, and then this was studied in a bit more detail. You see, we'll zoom into this area progressively more. So you see the cavity, and I'll call your attention to some of these basins or depressions. And so here is one such depression, and you see here a bit better, maybe you don't see it very well, but there's a bit of blue there to show bits of exposed ice, and this is part of the ice that sublimates. So here you see a pre and post burst outburst image of the same location. When Philae landed on the surface, they were expecting to see ice, and instead what they found looked pretty rocky. So a good way to look at the comet, at least that's what Jean-Pierre Biring likes to show, is that you have a mixture of ice seeded with organic molecules and silicates, and then you have sort of a sintered crust around it, which is the result of the sublimation of the ice, and then the other stuff stays behind. And it gives rise to sort of a, if you like, a cement, so to say, or a very hard crust on the outer part. Another thing that has been nice about Rosetta has been to follow the travails of poor Philae, who had a mission that was nothing like what we had planned in the beginning. So who can spot Philae here? It's there. So after a long time, finally, almost at the end of the mission, we are able to actually get an image where you could see Philae on its side. Another mission that has been very, very important for us has been Mars Express. I've tried to put only a couple of images in true color. I prefer to look at planetary images in the real color they have. Very often we end up coloring them for doing science or putting something more make it more visible. But these two I picked up here show Mars like it really is. It's not phosphorescent orange like you see many times. It's sort of boring gray-brown, which is something that is very important when you're on the surface. So here it's nice because you see the very thin wispy atmosphere and you see some of the clouds. And I will finish this tour of pretty pictures with a recent image from Akatsuki, which is the Japanese mission that is going around Venus now. This is actually not a true color image. It's taken with a UV filter that makes more visible the circulation patterns of the clouds. But they're doing very nice science with this. So I come now to a point that is important. Over the history of humankind, there has always been, religion is born out of a need to explain something about nature and about the universe and bring a bit of order to our lives. And religion has had many manifestations through the history of humankind, starting with the sun and the moon, and then the monotheistic religions more recently, relatively speaking. And science, to some extent, can be understood as an evolution of that. But there is a difference. In most religions, you are asked to believe something. There is some truths that are either contained in a book or delivered by somebody. The main difference between religion and science is that science starts by admitting that we are ignorant. And this is something that has been very, very powerful. We say we don't know, and we have a method, which is we propose an idea, and then we test it with experiments. But nothing is holy. At any time, you can say, ooh, this new experiment shows that what I believed before is incomplete or is not totally true. So this has served us very well, because as a result of learning more about what's around us, we have developed a whole bunch of technologies. But it doesn't mean that we are free from some of these dogmas or axioms or tenets. They're all somewhat synonyms. These are, how should I put it, truths that are accepted without really having a lot of proof. And in science, sometimes we see some of these beliefs that tend to be carried forward and have almost a life of its own. So I mentioned a few here. Oopa. So, for example, this idea that the universe follows a pathway of increasing complexity. Like, you know, we start from little things, we build them up into bigger things, we get to the organics, to life, and even that the highest expression of everything is a human brain. So this is a bit of a dogmatic or one of these axiom things that I think we have to dispel. Another one is even better, is that some people think that there is a unique pathway, which is also not true. There is a lot of randomness in the way things happen. Another one is that life is a necessary byproduct of increasing complexity. And finally, one that I like a lot is that water and life come together hand in hand. Now, as scientists who admit that we are ignorant, we have to get away from all this stuff and look at what it is that we have been able to prove. And there is not much that we can say. One is that life can be a product of increasing complexity, and that liquid water is an important requirement for life, but we should be careful to what extent we push these, if you like, these simple truths. So one thing we have been able to find is that things are amazingly bizarre in the universe, and that every new object we discover is nothing like what we had thought in the beginning. Even when we land on Mars and with all the images we have and the geological mapping and and we try to predict what it is we're going to find on the surface, time and time again we get surprises. And for example, Spirit, when it landed in Gusev crater, they were convinced it was going to be a crater lake and it was going to be full of sediments. Instead it was a sea of lava. And this applies time and time again to many of these objects. So, if you put that in the context of life, we have to ask ourselves whether life is exceptional or highly probable. There is talk about habitable worlds, but even this has to be handled with care, because habitable by what? We have seen that extremophiles can populate a large panoply of environments, but the conditions for generating life are very different from the conditions to sustain an organism that has already been created or appeared. And there are other conditions in which life can live, well, not live, can sort of protect itself and wait for better times, which are different from these other two. So it's habitable by what, habitable where, and habitable when. We heard about the concept of habitable zone, but as stellar systems evolve, that moves as well. And habitable does not mean inhabited. I mean, I can have all the ingredients and still not have life. So what we think we understand about the solar system is constantly being reworked by our findings with space missions, and indeed about what we find here on Earth as well. So when we try to probe the origin of life, there's two main ways of doing this. We can go backwards, typically by looking at biology, what we think we understand from cells, chemistry, genetics. The other approach is to go forward, and there we start with planetary physics and physics itself, things like circulation of fluids, how convection, real physics. And somewhere in the middle, these two things shake hands with geology. So I'll try to go a bit both ways, forwards and backwards, and the goal of this is to try to pick a few things that then we will use for space missions. So we mentioned that there are universal laws, and the collapse of a molecular cloud to form a solar system, that follows universal laws of celestial mechanics, things like gravity, exchange of energy from potential energy into heat, the way the sun evolves, the way the matter condenses in order, or accretes in order to form the first grains. But all of these can give rise to several different possible configurations, and ours is one, and as we have seen in some of the presentations before, is not even a very likely one. So we are on Earth and in our solar system rather atypical. So this shows that even though there are universal laws, the actual final product is as much a result of chance events as it is a result of these universal mechanisms. So, examples. The inward migration of Jupiter and Saturn. This is typical. It probably happens on most planetary systems. But the fact that it happened with the right timing and the mass ratio between Jupiter and Saturn to cause them to be re-ejected and seed the inner or the terrestrial part of the solar system with ice-rich planetesimals, we cannot say that that is typical. The collisions between planetary embryos, that had an effect on what the final mass, mantle convection patterns, atmosphere composition, and spin velocity and obliquity was for our planets. And that is chance. So for example, we got the Moon, and Mars remained small, but it could have turned any other way. So again, chance. Finally, for example, something that is very important for ExoMars is the way the infant phase of terrestrial planets proceeds. So after Alessandro Morbidelli stopped with these objects being accreted, that there is still an interesting phase there, and is a phase that defines what the initial conditions of terrestrial planets is going to be. I mean terrestrial planets, but this could apply to any other solar system as well. So we saw that the final stages of accretion of terrestrial planets was more violent and complicated than that of the gas giants. Why? Because the gas giants were playing cosmic pinball with a lot of material. So besides what is the bombardment, once you have that hot ball of plasma, sorry, a hot ball of magma, the sinking of the siderophile elements, the heavy elements, the radioactive elements, all the elements that love iron, iron itself, nickel, into the core of the planet. All that potential energy, as it went down, got transformed into basically heat. That liberated so much heat that the planets really were balls of molten rock. And this stuff loves water. So the water that came in with the volatile rich planetesimals was dissolved in this lava. Now, as this magma started to cool down, the ability to hold in solution volatiles decreased. So basically you started making soda water, so to say, in the magma. You had the bubbling out of the volatiles coming out. And this gave rise to the primordial atmospheres, which were hot gas envelopes, a couple of 100 bars in pressure. So hot that they were supercritical. Now, if you would have been on the surface of one of these terrestrial planets, you probably wouldn't have been able to see much, because no much light would have made it in. But basically it would have been bits of solid scum here and there floating on this big magma ocean. Something a bit like what you see here. And enveloping all this, you would have had these supercritical atmospheres. Now, supercritical fluids are very interesting. It's, imagine a gas that would like to be a gas, but the pressure is so high that it's being forced into being a liquid. And this is the way the supercritical fluid, you can understand, it cannot make up its mind whether to be a liquid or a gas. And it has very interesting properties. For example, we use it to extract caffeine from coffee if you want to have decaf, because it has different solvent properties. But these atmospheres radiated heat out to space. At a certain point, when you cross the critical point of water, you undergo a phase change. You know, in chemistry class, if I don't know, you're melting naphthalene in a test tube and you cool it down at a certain point, it all crystallizes in one go. So something similar would have happened here. The moment you cross the critical point of water, this water cannot stay up there as a gas anymore, and boom, it precipitates. It gives you a global ocean very, very fast. Of course, it wouldn't have been instant, because you have different distribution depending on the circulation patterns of the atmosphere, but it would have been rather catastrophic. So a deluge of biblical proportions, all this water raining down fast and giving you these global oceans, for example, in the case of the Earth. So this was still very hot water. Why? Because the pressure was still very high. It was like a pressure cooker. So the water may have gotten out of the atmosphere, but you still had all the CO2 and the nitrogen. But then things started to cool down. So initially you would have had Earth that was more or less a blue ball of water. There were no continents then. Maybe a few arcs here and there with a bit of land sticking out. And as things progressed, you started building up continental crust, and probably the first emerged lands appeared. So this view graph tries to put all of this in perspective. So on the top you see the various stages of planetary evolution. Here you see time, and here I've brought a bit, what we think are the oldest accepted preserved traces of life. And here you see the beginning of the solar system. You see the magma ocean, the condensation of water in the atmosphere, the emergence of the first lands. The interesting thing here that I want to call your attention to is that these infant planets were still hot. There was a lot of heat coming from the inside. So the hydrothermal activity during this period was massive. If you think of today as mid-ocean ridges and how you have crust being created, it would have been the same, but more or less everywhere. And we'll see that this is important. Mars, on the other hand, did not evolve exactly like the Earth. We know that at a certain point it took a wrong turn, lost most of its atmosphere and its water. But there is nevertheless two things that are important. One, we more or less know, give or take 100 million years, when this happened. And the other thing that is important is that the beginning was similar to that of Earth. You could say the same thing about Venus, by the way. Now, whether on Venus there was a liquid ocean or not, how long it lasted, that you have to play with that carefully, because it depends on how long the steam stayed, the water steam stayed in the atmosphere. If it stayed too long, then UV light would have been able to dissociate the water molecule, and then you would have lost the hydrogen, and you wouldn't have been able to condense an ocean. But okay. But in the case of Mars, because it was further away from the sun, and a faint young sun at that, it is physics dictate that you must have had large bodies of water condensing on the surface of the planet. Maybe not a global ocean, but a heck of a lot of water for sure. So this opens a window of opportunity for, let's say, life-compatible conditions, roughly 4.4 billion years ago, whether it's on Earth or on Mars. In the case of Earth, we know it lasted until nowadays. In the case of Mars, probably things started to dry up and become difficult on the surface, roughly 3.8, 3.7 billion years ago. This is a movie that was produced by the MAVEN mission, trying to show what Mars could have been like 4 billion years ago. I have some objections, but it still looks very nice. First of all, I think this cloud cover would have been thicker. And secondly, probably there would have been more water than what is shown here. Now, as the movie moves forward, they move forward in time. So you will see that the planet progressively becomes drier and drier. By now it looks very much like today. So, some people think that if you throw organics at water, you get your chicken soup. But this is wrong. I mean, you cannot just dilute organic molecules and expect that they will self-assemble and give you bunny rabbits swimming in the water. Things are more complicated than that. And so now I start to look at things the other way. So, poor Darwin, he's been shown so many times in these presentations. And what I'm saying here is not even his words. So people have tried to come up with definitions of life many times, and one that has lasted for a long time is this one. That was, I'm not sure who came up with it, but I know it's in the context of some NASA meeting. Life is a self-sustained chemical system capable of Darwinian evolution. It's elegant. I don't know if a mule, for example, you know, when donkey and horse get together would fit this thing, but from the point of view of somebody trying to put together a space mission, this definition is useless. I cannot do anything with it. How can I be expected to check experimentally Darwinian evolution when you need thousands of generations for something to happen? And the exchange of molecular information, catching the little beasts in the act of doing it, when you know, all I have is a crude microscope on the surface of another planet. So from the point of view of designing a space mission, I need something that is more practical. Maybe not as pretty, but something that we can use. And one thing that is interesting to me is, look at the people who are trying to synthesize life in the lab. So, you know, playing God in their flask bottles. They, you know, they're all over the world, and when they mix their things and they get something, they have to agree, is it alive or not? So, we saw a bit of this in Yuri's presentation. Maybe you cannot get a definition, but you can get sort of a set of functions that have to happen for something to be alive. Another interesting thing is, we know, somebody said life is like pornography, you know, I can recognize it when I see it, but maybe it's not such black or white. If something is not dead and then it's alive, maybe there is a gradual transition from being dead to being alive, sort of a progression, a gradual progression from elements to functional system. And I like this quite a bit, and so I found this other sort of definition that I think works better for what I need for a space mission. So, life being a network of mutually supporting chemical interactions that include the following functions, and we will see what they are. But the interesting thing is to go at this backwards. And is, we will need some functions, and they have to interact with each other. So if I have function A, it has to help what function B needs to do. So they have to work in a network, sort of like a system. So what could these things be? So one thing we need is a program. The program is a self-replicating information polymer that is able to catalyze other reactions. We already heard a bit about this. This polymer is formed of monomers. The monomers would have been assembled from precursors. It's very simplistic, but bear with me. And somehow these would have had to be harvested in one way or the other, we don't know, from the environment. So this is one function, program. Another important function, metabolism. I need to have the means to cycle between an energy-rich and an energy-poor condition. Today this is done with ATP, and then you carry your little electron and give it to whoever it is that needs it in the cell to do the other functions. Or you can do this with photosynthesis, but okay, here I put sort of a chemotrophic reaction. And this is this energy being passed on to somebody else that needs it. Again, this would have been synthesized from precursors who would have had to be assembled from something that was found in the environment. The third function is containment. So you need a self-assembling container where this stuff is protected and where it can do its all these catalytic functions between the various elements. So our minimal life form then, sorry, I forgot. Today this is, we deal with lipids to construct the membranes, and there may have been precursors for this, and they also would have had to be harvested from the environment. So our minimal life form basically consists of these three functions. If you have something that can do these three things, and you can demonstrate that it can do these three things, you have a reason to say, okay, this is something that is alive. But the story doesn't end there, because I'm trying to put together a space mission. So I need to know where I'm going to get this stuff. And then what I need to look at then is the environment. What sorts of environments could help me get this thing going? And so I need a few things. First of all, I need to be able to complex chemistry. That is, mean progress from something that is chemically relatively simple to something that is more complex. We'll get to what that means in a second. The other thing I need is I need some energy. So I need a proton gradient where I can, by some more or less complicated set of chemical reactions, I can harvest that electron that I'm going to pass to other functional groups in the cell to do the work. And the other thing I need is I need a slow flow of solvent. Why? Because I need all this stuff to be brought to me, and I need to get rid of what it is that I don't need. And it cannot be catastrophic flow. It has to be something that is reasonable. So if we dig a little bit more into this stuff, in the case of complexation, I need to be able to confine, fixate, and enrich. I admit this is super simplistic, because in reality I'm not saying anything with this. But imagine something that has to be offer a bunch of different conditions at molecular scale, because we don't know which one is going to work. In the case of a proton gradient, a way that works okay is if you have a pH gradient. So for example, acidic out and alkaline in. And in the case of flow, some of you may know what chromatography is, but that is more or less the same thing. You can think of a solid phase and a mobile phase. Okay, so finally, all of this has to happen in a relatively protected environment. It has to have mild temperature conditions. It has to be protected from UV, and this is very important. We've heard that some reactions are catalyzed with UV, but UV is a nasty beast. If you have a very wide UV spectrum and is very energetic, it tends to destroy more than help. And another thing that is very important, all of this is nebulous, vague. Most of it will never work. So if you want to get a chance that among all these things some will spark things right, you need to do this in a very variety of conditions in many, many places. The following conditions should have existed somewhere for at least a few million years to have the possibility to develop life ex novo. Now, I'm not saying that this is an exhaustive list, and you may disagree with some of these things. That's okay. I'm just trying to help the discussion along and then be able to draw something that I need for our mission. We need a UV protected environment, a mild temperature, a slow solvent flow, the ability to complexate chemistry, an environment that is ubiquitous and relatively stable. We need to have organic precursors, and we need a proton gradient. So where am I going to get all this stuff? Let's look at different conditions or different places. First of all, organics delivered to Earth, dropped willy-nilly on an early ocean. Which things work and which don't work? Is it UV protected? Well, I don't know, it depends. If you get them under the surface fast enough, maybe. Mild temperatures? Okay, you could agree that once the ocean has cooled down sufficiently, it's mild. Slow solvent flow? In some places, yes. The ability to complexate chemistry? No. I mean, if you have a dilute soup, okay, you may have some reactions between molecules, but you're not concentrating anything. Is it ubiquitous and stable? It's ubiquitous, all right. Stable, I don't know. If you had the moon close to the Earth and you had tides that were 300 meters going up and down several times a day, covering hundreds of kilometers going inland and then coming out, this was a pretty horribly catastrophic environment. Organic precursors, yes, because they're being delivered. And the proton gradient, not in the sea, because in the bulk of the matrix of the water, you basically have the same conditions everywhere. So, yes, you were delivering organics to Earth, but the chance that you would have had life develop in the bulk of the ocean, I don't think were very high. What about beaches? Beaches are great. I love beaches because it's like having a chromatographic column put there by nature for you. You know, you wash your solvent and then it just runs over the pebbles all the way back to the ocean. But it still has some problems. Is it UV protected? No way. The original Earth didn't have any oxygen, so there was no ozone. So, okay, the moment that most of the clouds started to thin down, it would have been a savage UV flow on the surface of the planet. It would have creamed anything organic that was exposed on the surface. Mild temperature, solvent flow, yes. Complexation, maybe. From the point of view of being able to run your solvent with a few organics down the beach. Stable, not very stable. I mean, you will get all this water rushing in and then going out and be exposed to very dry conditions and UV, and then later on during the day, boom, another inrush of water. And the proton gradient, not very sure you would have gotten that there. So beach, in spite of Christophe likes this a lot, but not so sure. Next one, subaerial hydrothermal systems. So hydrothermal systems on land. It's getting better. You have the problem of the UV, but mild temperatures, yes. You have low temperature hydrothermal systems, slow solvent flow as well, the ability to complexate, definitely. Ubiquitous and stable, I don't know. And organic precursors, yes. I'm not sure about the proton gradient. And we get to the last example, which is low temperature subaqueous hydrothermal systems, which I like a lot. Here, okay, I guess if you start looking at whether you have nitrates and phosphates. Maybe it leaks somewhere, this. But from this very simplistic picture I tried to paint, this definitely ticks all the right places. And there's another thing that is beautiful about this, that this thing that you see here was replicated gazillion, billion, million times all over the crust of the Earth. In the very beginning, you had water everywhere, not much continental crust, and these things all over the place. So if you can think of the entire crust giving birth, so to say, the same experiment in organic chemistry was being repeated with slight variations all over the place. So, and how did it work? Well, in the very beginning, you would have had slightly acidic oceans, and you would have had the sequestration of organics, the initial sequestration of carbonates by reaction with oxides of the basaltic crust being presented to the ocean. Then you would have had cracks, infiltration of water into the mantle, and this water that goes down then reappears in the form of an interesting exhalation in the hydrothermal vents, because on the way down it leaches a whole bunch of elements and acquires some others. And in particular, the reaction, the hydrolysis reaction of water at, let's say, 100 to 200 degrees with ultramafic minerals gives rise to the release of hydrogen. And what comes out here is very reducing. So you get to hydrogen, methane, formaldehyde, methanol, a whole bunch of stuff that comes out of these hydrothermal vents. Going through this micrometric porous structure where you have temperature gradients, you may have 100 degrees or 80 degrees through the main chimney, but then as you move to the left and right, then your temperature drops down. And as these organic molecules go in, they encounter all these interesting minerals, some of which have catalytic properties. So they're happy to stay in there. And you have this continuous slow flow of organic rich water going through the system. So it sort of takes care of bringing stuff and taking away stuff with it. So it's an interesting environment. So roughly something like this. So you're going with your soup, your organic soup, through a mixture of different minerals. Admittedly, this is pretty simplistic. What it tries to show here is you go in with simple things and you come out with, something in vesicles. It's a bit, maybe a bit too simplistic, but that's the idea. Now this would have happened very readily. The reaction of ultramafic basalts with water would have given you hydrogen, and this is also one of the keys to understand why the early Earth was not a snowball. Because even though the sun was weaker, it was only shining 70%, and therefore the temperature on the surface should have been something like -30, -40 degrees. This process gave you a lot of methane. But you noticed in that view graph I showed you how the hydrothermal activity would go down with time. So this gave you the additional methane you needed in order to force greenhouse effects to bring the temperature of the planet above the freezing point. Today we get 35 degrees of greenhouse forcing from what we have. In the past, we would have needed somewhere between 50 and 70 degrees of greenhouse forcing to stay above the freezing point. Well, it's not a lot to ask. You're asking for an additional 15 or 20 degrees of greenhouse forcing. And this process not only helps you along with the, let's say, the chemistry that may be interesting for the origin of life, it also helps you to warm up the planet in the very beginning. And luckily for us, as the sun started to increase its luminosity, this activity started to wane down. So it's a very nice confluence of things. So let's look for life. You have a bunch of options. You can search in a place where life could have developed. You can search in a place where life can or could have flourished. What does that mean? It means I have a cell, I have a microorganism, and it's able to live there. Or you could search in a place where life could have collected. This is the one I like the least. That is why I don't like to go to riverbeds or lakes or all this stuff. Deltas, I hate them. Because these are places where you get detrital organics. I mean, the stuff was maybe alive somewhere else, and then it died, and the stuff is brought to a place where you can collect it, but you don't know how it appeared, you don't know the environment in which it grew, you just get the detrital signatures that may have been live somewhere else. Now, the conditions for life to appear and for life to flourish are very, very different. It's much easier to keep a microorganism that is already functioning alive than it is to create the first cell. There is a much larger distance between prebiotic chemistry and the first cell than there is between an amoeba and you or I. The cell is a very complicated piece of machinery. So I would posit to you that all other things being equal, you're much better off going to a place where you think life could have appeared than to a place where you think the conditions would have made it habitable. Now the question then is, can you get there or not? Because this complicates things a great deal. So here's a few types of environments. Obviously, if you can land here, that's very interesting. But this is not easily accessible. Landing here doesn't help you a lot. This is not too bad. And for example, in the case of Europa, it would be very challenging to be able to say something. Enceladus I like quite a bit because the moon is serving it to you on a tray by having these plumes and sending the stuff out. That's a really cool mission to do. First of all, the concept of habitable zone. It's not sufficient to say there is anywhere where liquid water is stable. We have seen now that you need much more than just liquid water. Liquid water, yes, is necessary, but you need a confluence of other things. And some of them are not easy to verify because they happen at a small scale. Another thing, the solar system is not very typical, as we saw in Alessandro's presentation. The delivery of water to the inner solar system may not be something that is very general. We saw in Charles' presentation that life can colonize many extreme environments, but that doesn't mean it can appear in many extreme environments. So you have to be careful about that. And finally, it may be that the conditions we have on Earth, or if you like in our terrestrial planets in the beginning, are not that typical, but that the submarine hydrothermal conditions of Europa and Enceladus are more typical in other solar systems. So, why life on Mars? Why do people keep on beating this drum year after year of life on Mars? Well, first of all, the early evolution of Mars was very similar to that of Earth. So some of the processes we believe to have been important for the appearance of life on Earth could have happened, or probably happened, on early Mars as well. For example, the presence of liquid water. But very importantly, you heard in Christophe's talk how we don't have access to very early rocks on Earth, let's say 4 billion years of age, that are in a good state of preservation. On Mars we do. We know where they are. We can see them on the surface. And because Mars hasn't had any plate tectonics, or at least global plate tectonics pattern, they are there as the way, in the same manner as when they were made. Give or take a lot of meteoritic bombardment and burial by other materials. But in theory we should have access to very ancient rocks with a very low degree of metamorphism. So this is great. Let's go to an ancient plains of Mars. And Mars could have been something like this. This is taken from one of Jean-Pierre Bibring's presentations. So today, however, Mars is a horrible place. It's a cold desert bathed in large doses of UV radiation and ionizing radiation. And we have to deal with the question of the early habitability at no longer planetary scale, but at landing site scale. And also another thing that is very important is the preservation of these possible biosignatures. How did they make the time travel from 4.2 billion years ago until today? Will we be able to recognize them if we see them, or will they have been messed up so much that we cannot tell anymore if they are biosignatures or not? We said that life needs, more than anything else, liquid water. And this is indeed important. But liquid water is no longer present on the Martian surface because it's unstable. The pressure is just too low. So if we wanted to look for life on Mars today, we would have to look in the subsurface, deep, like 2 kilometers deep, because there the vertical column of minerals you have on top of you give you sufficient pressure to have liquid aquifers. So in theory, if we had life on Mars in the very distant past, it could have retreated to the subsurface, particularly if it was chemotrophic and didn't rely on sunlight. It could still be doing its stuff in the subsurface deep. The other thing is, say, well, let's look for traces of extinct life. We cannot look for traces of extinct life close to the surface because there it will have been messed up by ionizing radiation, UV light, oxidants. So the trick is also there. You have to find it preserved in the geological record, but somewhere where it has been protected. Now, life is cells. That's a very easy definition if you're trying to put together a space mission. I don't worry about any definitions, Darwinian evolution, blah, blah. Cells. If I can find cells, great. Now, if it is ancient life, however, I cannot have access to cells. I can only have access to what remains of the cells. So here we say, if it is present life, I can target any of the Lego building blocks of cells. Sugars, phospholipids in the membrane, amino acids, nucleobases, If I can detect any of these in a reasonable state of preservation, I can say, oh, I have something that is either alive or has died not so long ago. In the case of past life, particularly very ancient life, it's unreasonable to think that you're going to find molecules like this. You're going to find out what remains of them. So some of the functional groups may have been lost, but for example the carbon chain may be preserved. You have to be a bit of a Sherlock Holmes. And whatever you will find will be complicated by the presence of delivered organics. There are several hundred tons of organics that have nothing to do with life that fall onto the surface of your planets every year. So We haven't been the first to search for life. In the 70s, the Cadillac of the skies was sent to Mars, a mission that today would have been 10 billion plus. Two rockets with 2 satellites and 2 launchers, the Viking, and 2 landers, the Viking missions. The first images were amazing. I mean, look at all this stuff. Rain or snow on Mars. You can see here flow on the surface. Evidence that there was buried ice. You see these low-bade craters. Here's the crater on the moon. Boring, just dry stuff. Instead of Mars, big splash. The first images of the surface. There was even frost on Mars, condensing during the night. And the first search for life, which was digging trenches at both lander sites and then analyzing the stuff. Now, these guys were extremely clever. You wouldn't believe how much effort and care went into preparing the instruments on the Viking. So there were 3 biological instruments designed to detect life, but these guys, they were convinced they were going to find life on Mars. So they went for detecting metabolism of cells on Mars. And as sort of an afterthought, they had a gas chromatograph mass spectrometer to look for organic molecules. But that was not conceived as a life-seeking experiment. They were actually trying to find the meteoritic organics. They knew there would be meteoritic organics, and they thought when they landed on Mars, they would have 1 or 2 meters of organic stuff, black, mixed in with the soil. So they were prepared to detect these meteoritic organics. Now, these experiments were very, very smart in their simplicity. So, for example, I have to read this because I never remember to get everything right when I go through them. The first one, the pyrolytic release, what they did was to cultivate a soil sample with light in a simulated Martian environment. Now, they use radioactive C14 because in that way they wouldn't need any fancy equipment. All they needed was a radioactive detector able to say, I can detect radioactive carbon. So they cultivated this stuff for a few days, removed all the gases, pyrolyzed or heated the soil, and they knew that if they detected any carbon-14, it meant that it had been taken by the bugs from the atmosphere onto the sample. The second one, the label release experiment, was also extremely interesting, also worked with carbon-14. They prepared sort of a very nice soup full of sugars and water and fed it to the ground. And then waited and monitored this time the atmosphere, looking to see if carbon-14 would be released in gaseous form as a respiratory byproduct. And the final one, they didn't use the carbon-14 for this, but it was more classical, if you like. They put the soil sample, they added an artificial atmosphere with a water-based nutrient, they let it stay there for a number of weeks, and then finally they cleaned everything, pyrolyzed the whole thing, and this time they looked at everything that came out. Now, these guys were so smart that they realized that they could get an ambiguous result. So they say, we'll try to remove ambiguity. So they also introduced a means to eliminate false positives. So they also heat sterilized the samples to 600 degrees and then conducted the very same experiments. And this, by heating them to 600 degrees, they knew they were going to kill anything that was alive, and then they expected to have the results that you see here. So in the case of life, they would have expected responses on all 3 experiments and no response in the heat sterilized one. And in the case of no life, they would have seen the same answer on both types of experiments, the ones conducted at life benign temperatures and the one conducted at, after they had cooked the sample at 600 degrees. And this is what they got. If they hadn't had another instrument to sort of solve the impasse, some of them detected life by the standards they set for themselves before the mission, because they had reactions. Imagine they hadn't done this. They had only done that. They would have had to conclude there is life on Mars, and it's happy and it's metabolizing. Because they also got some emissions with a heat sterilized control where they knew there would be nothing alive, then they started to look for an explanation that involved non-organic chemistry or no life. And the one that solved the impasse was a gas chromatograph mass spectrometer that failed to detect any organic molecules. So how could you be having this stuff being alive if there were no organics? So in the end, the prevalent view, although there are some people that still feel that Viking did detect life, but for the most part, the failure to detect organics on the surface of Mars sort of sealed the fate of whether there was life on Mars or not. And today we have moved on, and we think that the negative results of Viking do not preclude the possibility of life on Mars. But definitely this put a stop to Mars exploration for almost 20 years. Mars all of a sudden became not a very interesting target anymore. And for many years NASA stopped with missions to Mars. So out of this scenario this is the cradle for the way ExoMars was born. It was in 1996, ESA assembled a group of international scientists and told them, based on what we know from Viking, if you wanted to go look for life on Mars today, then, how would you do things differently? And this will be a bit the second part of the talk. Before I finish with this, I want to go through very quickly through a couple of other important missions that have helped us with our understanding. One is Mars Express. I don't mention it here because the other ones were not important, but one of the very important things that had been lacking for a long time was where were the clays? Because if there had been water very early in the history of Mars, when water reacts with basaltic material, clays are formed. And why didn't we see any clays? And this was one of the major discoveries of Mars Express, the presence of clays, and in particular the presence of clays in association with very, very old terrains, not with the modern ones. So all this history about water having gone away and the dry period, it's all out of this little mission that was put together with the instruments that remained from the failure of Mars 96. Another very important mission for understanding Mars, Phoenix. Phoenix landed in 2008, and for the very first time it tried to do something different. It had what is called a wet chemistry lab. Doing wet chemistry on Mars is a pain because the pressure is so low that water sublimates. So they had to pressurize a cell. And every time you try something new, you find novel things. And in this case, they found the presence of this very strange anion, which is the perchlorate anion. Perchlorate is a pretty exotic salt on Earth. Typically you form it naturally only on high altitude deserts or where you have a lot of contamination, like in Los Angeles, for example. What you need is a source of chlorine. Typically this can come from volcanism, and very high doses of UV light. But the interesting thing about perchlorate is, you see the structure here. You have the chlorine in the center of a tetrahedral molecule with 4 oxygens. Perchlorate is stable. It doesn't do anything until you heat it up to a couple of 100 degrees, at which point it liberates the oxygens, which are extremely powerful oxidation vectors. So when they saw this, somebody had the good idea of going back to the Viking results. And Viking had detected chlorinated molecules, chloromethyl and dichloromethyl. And at the time, they thought these were the results of the detergents, or the cleaning agents they had used to clean the spacecraft. But what is it that is really happening? You put your sample in the oven, which is mixed in with perchlorates that are natural on Mars. You heat it up, the perchlorates dissociate, the oxygen that is liberated oxidizes the organics that may be there, so you're destroying what you want to study. And the chlorine, poor chlorine, he says, I also want to connect with somebody. So he goes and finds the simplest organic he can find, methane, and boom, bumps off an hydrogen and I'll chlorinate you. And you get chloromethane or dichloromethane. And this was not a peculiarity of Phoenix, but has also been found by Curiosity, which has had its own organics measurements complicated by the presence of perchlorate. So we'll explain later on how we'll deal with this. So there's a whole bunch of missions being prepared. I'm showing here only the ones in ESA. And just finished this part with a couple of slides of another mission I think is super cool besides ExoMars, which is JUICE. Now, JUICE will go to the Jovian system and it will make a couple of flybys of Europa and then settle into orbit around Ganymede. I think this is also a very interesting mission, and it's interesting first of all because of where we're going, but also because of the super cool instruments that are being developed. So this is the end of the first part, and on the second part I will talk a bit more about ExoMars and how a lot of the things we have discussed first are put together on trying to devise a mission to search for life on the surface of Mars. So sorry I ran over by 5 minutes. Okay, thank you.